In this episode, we'll be exploring the concepts of mutagens. Mutagens cause changes in genetic material and consequently cause a mutation within the DNA that can be carried on to the offspring. Not all mutations are bad. Some form the basis of evolutionary change within a population due to the changes in the phenotype. However, some mutations can have very negative consequences for the organism. Today we're just going to be looking at mutagens that cause mutations. We'll be exploring electromagnetic radiation sources, chemical sources, and naturally occurring mutagens. Recall back to our understanding of protein expression in protein synthesis. We know that DNA is transcribed to form mRNA, which is then translated into polypeptide chains that is then used to form proteins for cell structure and function. Mutagens may be physical, chemical, or biological. We're exposed to mutagens on a daily basis. However, our DNA repair system are usually able to detect changes in the DNA and fix errors. Mutagens become a problem when the repair system is compromised and may lead to diseases such as cancer. Electromagnetic radiation are emitted by electrically charged particles. These waves are able to interact and ionize with atoms which they encounter in the body. Radiation is highly penetrative because it is composed of high energy waves. This means that they can interfere with the DNA molecules in the nucleus. This interference bonds with the DNA structure and cause it to break. This causes chemical composition changes in the DNA molecule. This can result in deletions, rearrangements, or shape changes in the DNA. Due to the high energy states of the radiation, they are able to disrupt the hydrogen bonds between the nitrogenous bases, as well as the bonding of the sugar phosphate backbone. Examples of electromagnetic radiation include gamma rays, X-rays, and ultraviolet UV light. Radiation can damage DNA indirectly by ionizing another molecule in the cell to produce free radicals. Free radicals are highly reactive, and as they have unpaired electrons, this means that they strip electrons from any molecule they encounter. This includes DNA. Let's look at an example. UV light is a form of radiation that we're exposed to every day with the sun. UV is damaging in high doses. DNA exposed to high-dose UV form a structure called a pyrimidine dimers. This is when the adjacent nucleotides form a covalent linkage when excited by the energy. In Australia, the average rate of exposure to UV produces 20,000 pyrimidine dimers per hour. That's a substantial amount of DNA that needs to be repaired. Unrepaired DNA results in DNA replication or transcription of a gene that can result in disease such as melanoma, known as skin cancer. Chemical mutagens interact with and have mutagenic effects on DNA both directly and indirectly. There are three main types of chemical mutagens. We have radioactive agents that release radiation such as alpha particles, beta particles, and gamma waves, and these are able to penetrate the cell and interact with the DNA bonding and create disruptions to the DNA. We also have intercalating agents such as ethidium bromide, and this chemical causes a frame shift mutation during DNA replication. This means that they're highly carcinogenic. This chemical is used in labs to observe DNA more clearly. Metals such as arsenic, nickel, cobalt. There are several different mechanisms that metals cause mutagenesis. They can affect DNA repair, causing the base mismatching and errors with the correction of these errors. This process can also cause the errors to be passed on to daughter cells. Nickel, for example, inhibits the ability for histones to condense, affecting the chromosome formation of DNA. Naturally occurring mutagens include viruses. They replicate by inserting their DNA into host cells. This creates a disruption in the normal cell function, and this may lead to lasting mutational changes. We have bacterial infections can cause inflammation, which reduces the efficiency of DNA repair, that therefore increases the rate of mutation. Then you have transposons, referred to as jumping genes, are segments of DNA which can change their position in chromosomal DNA. This directly alters the expression of genes in cells. This phenomenon is related to those examples of peppered moths and antibiotic resistance in bacteria. From this clip, you should be able to answer the inquiry question, how does a mutation introduce new alleles into the population, by referring directly to mutagens. You need to be able to describe electromagnetic radiation using some examples and explains how it is effective as a mutagen, the same for chemicals and naturally occurring mutagens. See you next time, budding biologist.